afternoon, everyone, and welcome to today's Public Health Ontario's Microbiology Rounds presentation on developing a genomic surveillance program for syphilis in Canada and Ontario. My name is Angela Ma. I'm a clinical microbiologist at Public Health Ontario, and I have the pleasure of moderating today's session. Before we begin, I will mention a few housekeeping items. The chat pod has been disabled to limit any distractions during the presentation. We request that the audience please use the Q&A pod if you have questions during the session, and a discussion and question period will follow the presentation. If at any point during the session you experience any technical issues, please email capacitybuilding@oahpp.ca. I would like to state that as the moderator of this session, I do not have any conflicts of interest. It is now my pleasure to introduce the speaker for today's presentation, Dr. Tom Brockman. Tom Brockman did his PhD at the University of Toronto in ecology and evolutionary biology. Following his PhD, he worked as a postdoctoral fellow at the University of Guelph and then at Stanford University. He joined PHO in May of 2022 as a scientist in genomics and bioinformatics, and he currently works on projects involving SARS-CoV-2, Mpox, Mycobacterium tuberculosis, the causative agent of tuberculosis or TB, and Treponema pallidum subspecies pallidum, the causative agent of syphilis. Tom, take it away.
Thank you for the introduction, Angela, and thank you everyone today for coming to hear about my talk on developing a genomic surveillance program for syphilis in Canada. I would just also like to acknowledge the other authors. So a lot of this work is done in collaboration. So on the PHO side of things, Dr. Venkata Devuri, Dr. Vanessa Tran, who I've been working a lot with. And then at the NML, we've been collaborating with Dr. Raymond Singh, who's leading a GRDI to help support this work. And I'd also like to acknowledge Mackenzie Neal, who's done a lot of the lab work for this. I'd also just like to say that standard disclaimer that this work is my own. PHO has no liability, and I'd also just like to state that I have no conflict of interests. So today, it's going to be in a slightly different order than what's in the prelim talk, but I'm going to talk about the epidemiology of syphilis in Canada and abroad. I'm going to talk about the Treponema genome. So syphilis is Treponema pallidum subspecies pallidum, but talk about the challenges of sequencing Treponema genomes, particularly the pathogenic ones. And then I'll also talk about how we can go about sequencing Treponema pallidum. So before I start and usually get into things, I like to kind of talk about definitions. And since we'll be talking today a lot about metagenomics and genomics, I think it's very good to talk about how I define them. So genomics actually has a lot of definitions. I usually prefer the most inclusive, simplest definition. And genomics is simply the study of an organism's full complement of genetic information. This can be from looking at genome structure and function to kind of population genomics within a species or among species. You can think of it as looking at the evolution of antimicrobial resistance. And then as a general rule, all genomes are DNA-based with the exception of viral genomes. So metagenomics also has quite a few definitions, and it's a fairly broad subdiscipline, but again, it's really the study of genetic information from samples containing multiple organisms and or viruses. It can be some things looking at diversity within a sample. So if we think about looking at microbiome, so looking at the species diversity, but also kind of what a community is capable of doing. So the ecological niche, so what metabolites are there is metagenomics. And commonly in public health, so if you think of the Mpox outbreak, for example, for the first while, a lot of people before an amplicon-based approaches were developed. Sequencing all the DNA from a patient sample to kind of capture a targeted pathogen genome. And so for the purpose of the talk, we'll just call, when you have a patient sample, we're just interested in a particular pathogen, we'll call it targeted metagenomics for the talk. The literature can sometimes refers to that as 16S genomics, but for convenience today, I'll call just sequencing a pathogen from a mixed sample targeted metagenomics. So when we talk about Treponema, the genus itself has pathogenic and non-pathogenic species. They are spiral shaped, gram-negative motile bacteria. And of particular interest is Treponema pallidum. It's the pathogenic species, and it's actually comprised of 4 disease-causing subspecies. So we have endicam, which causes endemic syphilis, sometimes referred to as bejewel in the literature still. We have Pertenu, which causes yaws, caratium, which is a causative agent of pinta. Yaws and pinta, for example, cause skin conditions. And then we have venereal syphilis, which is caused by Treponema pallidum, subspecies pallidum, which I'll just refer to as pallidum for the rest of the talk. Which causes the SCI that we're most more familiar with, and that's going to be the focus of our genomic approach. So when you actually look at these 4 subspecies, though, they all look very similar. So they have a very similar morphology, and they're actually often antigenically cross-reactive. But they do differ in their epidemiology, how they present clinically, and where you most frequently see them. So, for example, pallidum is circumglobal, whereas other ones, such as can be more regionally localized. And so genomics itself can be challenging for sequencing any of these species because they're not readily or easily cultured. So, for example, if we look at syphilis, you can technically culture it in rabbit testes, but that's not a very efficient way to go about sequencing genomes readily. So there has to be ways to circumvent it. And so part of the challenge with is that, but when we actually look at the genomes of these 4 subspecies, they actually share a lot in common. So not only are they morphologically similar, they're actually very genomically similar. So all these 4 subspecies share about a genome that's 1.1 megabases long. And more interestingly, they have a very similar gene content. So their core genome is very similar across them. We do see differences in gene contents, and so in these genomic and pathogenicity islands, let me just get the pointer here. So these here, you have a genomic island and these pathogeny islands, they differ among the 4 subspecies. So what's interesting about Treponema pallidum as a species is that relatively small changes in gene content can lead to completely different disease phenotypes. When we just look at subspecies pallidum, what we do know is that the genome is very conserved across everything that's been sequenced to date. Relative to its other subspecies, it does have some unique genes relative to like amino acid synthesis, lipid synthesis, and other things. And that the genome itself is 1.1 megabases. And what makes pallidum such a good candidate for genomic epidemiology is that it is actually relatively very slow evolving. So if we actually want to kind of look at how syphilis is spreading and how different strains are related, using a few markers is going to underestimate that diversity because you can have relatively small changes. So I think one paper they had observed no change in some strains over a period of 2 years. So those are makes it a good candidate for genomics because you can use all the genomic information to capture that. And more importantly, within what we can use genomic for as well, there are what we call these Treponema pallidum repeat proteins. These genes are paralogous in the genome, so there can be some challenges with sequencing them. And then they produce surface proteins. So there's adhesions and porins that vary among closely related strains. And so what makes these genes particularly interesting is we can use them as targets for vaccine development or for development of point-of-care testing. So that's kind of something we'd hope to do is to kind of start understanding the evolution of the pallidum genome among all these closely related stains to kind of do that, to track that. And more importantly, what we can do as well is we can use genomics as well to start doing antimicrobial resistance stewardship. So we do know within syphilis genomes, for example, or pallidum genomes, that there's macrolide resistance is relatively common. It's not fixed, but it is common. And then just a fun fact that I always think is cool about syphilis, the it is capable of coating its cell surface with host serum proteins or polysaccharides to avoid immune response. That's just more fun fact for me. And I think we have another poll question. Yes. So just kind of, what's everyone's answer? Yeah, so I'll talk about this. It's women have seen the largest increase, but we see increases across the board. But we'll talk about that. Yeah, so so when we actually look at the diversity of syphilis, is everyone right? There are two sublineages. So you have the if you this large clade up here, this is the SS14 lineage, and then the Nichols lineage. They diverged from each other quite a long time ago. We do see more diversity in the SS14 lineage, but both of them over the last 20 years have undergone recent expansion. So as cases have increased, we're actually starting to see more diversity in both. And what we actually do see is that many of the sublineages you see within either Nichols or SS14 are shared across multiple countries. So usually whatever sublineages we do see, it's rarely exclusive to a single country. And so this is actually a kind of a fascinating case, is because we do need genomic surveillance to kind of understand what is going on with these transmission networks and patterns to see what is shared globally. Are there strains that are exclusive to a country, and kind of understand these dynamics. And so a good example of what how useful genomic epidemiology can be is in the UK, they sequenced 237 Treponema genomes from 2012 to 2018. They did notice that a lot of the strains that they saw circulating in the UK can also be found elsewhere globally. And this is probably because syphilis is so slowly evolving, you'll usually find closely related things. And that the two most dominant sublineages that they saw, one was being SS14, one being Nichols, were also seen circulating globally as well. And that they noticed that a lot of particularly the ones that were the dominant sublineages, were resistant to macrolides. So when we look at these two sublineages, what is actually interesting about them is that, well, one is belonging to SS14. The epidemiology differs between them. So sexual orientation, for example, the SS14 cluster was spread among men have sex with women, gay, bisexual, men have sex with men, women have sex with men, more so than the Nichols lineage, which tended to be more restricted or more commonly seen among gay, bisexual men who have sex with men. They did not note it. So there was previously stated like a strong correlation with HIV status and syphilis. That wasn't strongly observed in the data that they had, which is interesting. So that's a change from previously observed. And then they did notice for the SS14 lineage, it tended to be more evenly distributed among different age groups, whereas the Nichols lineage tended to skew more strongly among older individuals, or 35 and up, which is not really older, but anyways. Yeah, so that was kind of interesting. And then more importantly, when you actually look at the geography of it, how these strains were distributed across the UK differed. So if we look at the Northeast, for example, they had predominantly SS14, and then they had these two clusters, and particularly this one cluster here on the right was a set of cases in the UK that most strongly was very closely related to a sequence from Hungary. And so this, for example, this shows a potential case of importation of a syphilis strain into the UK, followed by local spread, whereas other clusters were not so clearly delineated among those events, having that thing. Now, when we look at Canada, what is going on in Canada is that it's becoming an increasingly public health concern for syphilis in Canada. So in 1998, the national goal was to have a half case per 100,000. In 2020, there were 24.7 increases. This increase was most particularly prevalent among women or females, as they saw a 700+ plus% increase from 2016 to 2020. And if we actually look at Ontario, this trend is holding true in Ontario. So there were about 15.9 cases in 2020 and about 19.9 cases in 2021. So we're still seeing increasing rates in Ontario. And we're actually getting closer and closer to pre-penicillin rates, which is shocking because penicillin is still an effective treatment for syphilis. So it'd be genomic and genomic epidemiology would be a great way to kind of understand what's going on. Within Canada, we see that trend that we see globally is that rates are rising in both men and women. It's just more that rates have dramatically increased in women, particularly from 2016 is when we start seeing this rate increasing quite a bit. And the challenge with this, as many people are familiar, is we're starting to see more congenital syphilis. So it would be, there's kind of a need to kind of understand what is circulating in Canada, where it's circulating, what strains are, do we see any like common patterns to kind of help understand what is going on with the increase in syphilis. So part of the goals of this program that we've started in collaboration with the MNL is to kind of develop, is to kind of implement a syphilis-specific metagenomic analysis of clinical specimens and kind of a standardized whole genome sequencing approach and pipelines associated with that, so that can be readily deployed by public health labs across the country. We'd also want to kind of use this genomic information, and so this is a collaboration with the NML to develop novel point-of-care tests. And more importantly, we're also interested in conducting the kind of molecular genomic surveillance, kind of understanding outbreaks, the kind of spatial temporal dynamics that you see with syphilis. And more importantly, also, not more importantly, but equally importantly, is to establish kind of antibiotic resistance, susceptibility prediction and detection for good AMR stewardship, or antimicrobial resistance stewardship. So the way we kind of envision this program working, and this is funded through a GRDI, is that we will, once it's up and running, we will receive samples from BC, Manitoba, and Ontario. And then these will undergo sequencing. And then these genomes will be combined with epidemiological data, which can then be used in this objective 2, which Venkata is leading, to help develop broad point-of-care tests. And then what we can also do is we can do antimicrobial resistance surveillance. We can also use these genomes combined with epidemiological data to kind of understand, to outbreak modeling and understand the epidemiology behind the increase of syphilis in Canada. So objective 1 is the first challenge. So as I previously stated, there's not an effective way to culture pallidum in the laboratory. So it's essentially non-culturable for our purposes. So we have to go about using metagenomic approaches. And so the way we're going about validating our lab methodology is that we're going about testing a few different ways of going about it. So one way in which you can go about enriching in your pathogen and depleting your host, because just like viruses where it's the genome small relative to the massive, like a 3 gigabase per human genome, your sample is going to be overwhelmingly human DNA. That's the same thing with syphilis, because it's a relatively low parasite load, that whenever you just sequence a sample, you will less than half, like 0.5% of your reads, or less than 1% of your reads will be actually your target, which is in this case pallidum genome, pallidum. So what we wanted to do is we wanted to compare a bunch of different methods to kind of select the best workflow. So we're going about comparing no host depletion with a couple host depletion methods. So I'll explain what these are. And these host depletion methods really clump into 2 different methods. So you have one which is a host depletion that's kind of post-extraction, where and then these other ones which rely on, I call differential lysis. And then we also wanted to look at host and target enrichment using what we call specific whole genome amplification. So it requires an isothermal amplification that's biased towards your pathogen. And then we also wanted to look at bait capture, which is also known as hybrid capture or hybridization bait capture, to kind of see how well that will enrich for a relatively low pathogen with relatively low parasite load. So when we look at these kind of this host depletion, so one of these sample biasing methods, when we think of differential lysis, what you have is when you have a sample, we have human cells here in blue, and then we'll call these bacterial cells or syphilis cells in these little green eyes in your sample. And so when you go about this differential lysis, what you do is, because bacterial cells are a little bit more robust than eukaryotic cells, you can just selectively lyse them using osmotic methods. And so what this does is it creates you have free-floating host DNA and intact bacterial cells. And what happens then is you can use is what QIAGEN does, or the QIAGEN kit does, you can also do yourself, is you can then use DNAs to digest or remove the host DNA. The LiPMA method that I had previously stated, what that does is that chemically inactivates the host DNA, so it effectively removes it from downstream processes. And so what this relatively leaves you with is just a fraction of your sample that is heavily enriched in bacterial cells, which you can then extract and use for downstream processes. We haven't been able to test this approach because one of the things that we're doing to optimize, and I'll share data from today's cells cultured in rabbit testes, but even some of the initial clinical samples that I'm waiting on the results for, they're all pre-frozen. And so the problem with this method is it requires material that's not pre-frozen, relatively fresh, because if you freeze and you have crystalline structures that all this is going to be lysed, then you won't be able to achieve to separate or everything will be lysed by the time you get it, so you'll just digest or inactivate all your DNA. That's temporarily on pause. And whereas the selective enrichment, which is that NEB has a kit for, which is in this case you have your sample, you just do a total gDNA whole cell extraction. The way this approach works is that eukaryotic DNA has higher rates of GC methylation. So you have these proprietary beads from NEB that basically is able to bind that GC methylated DNA, and then you can selectively pull that out of your sample, which should hypothetically leave you highly enriched with your pathogen DNA. And then you take that forward to sequencing. So we did test this. And then when we actually want to look at our target enrichment methods, there's two approaches. So you have selective whole genome amplification, which is just a modified whole genome amplification. So the method is the same for both. And the way this method approaches, in the case of selective genome amplification, you design these very short primers, usually 6 to 8 base pairs, but it can be up to 10 or 11 base pairs that match or target against your target genome. So this in case the pallidum genome. Since these are relatively short, there's a chance that they'll also amplify the host, but the idea is that they'll be preferentially binding your target. And so what you do is we'll actually undergo with isothermal amplification using Phi-29 DNA polymerase. What that does is once you start doing an amplification step, you got these long strands, and then they'll basically start displacing when they run into another strand. So you can actually generate very long fragments of DNA. So kilobases long, many kilobases long. And then what will happen is these primers or these hexamers, octamers, or even long decamers will continue to bind, and it'll just keep creating these fragments of varying length that can get up to very multiple kilobases and long. And then you would take this forward into your library prep and sequencing. And the advantage of this approach is that it's very compatible with nanopore. So you can just take your sample, do your amplification, and then run through your nanopore library prep, for example. And then another thing is bait capture. So with bait capture, you just extract your total DNA, you shear it to an optimal size. And what happens is, and I hope everyone can see this, is what happens is prior to your bait capture step, you basically bind these baits that are RNA molecules, biotinylated RNA molecules, that are 120 base pairs in length. And they're designed across your target genome. So you can create a pan genome or just take a reference genome, and you create these 120 base pair mers that are evenly spread throughout across the genome. And what they do is they bind to your target and then they pull it down. So they bind to your target DNA. So they're biantillated. You can selectively pull these out with a bead capture that will preferentially pull out these biantillated baits. You still get host capture carry through, particularly in our case, because host DNA is so much more prevalent than the pathogen DNA, but you should end up with a very highly enriched fraction of pathogen DNA that you take for sequencing. So we did test these methods, and so one thing I've been wanting to do with our nanopore data, which I won't present today, is assemble de novo genomes using a combination of these approaches. But today I'll present data from our control samples, and it's relatively simple pipeline where you have your raw Illumina sequencing, which I'll present today. You filter out your low quality reads, you map, and I did a composite mapping. So you map your reads to a reference. And so in this case, the reference is the human genome plus a syphilis genome. And then you just take the reads that map to the syphilis genome to avoid contamination from your host. And then this was moved forward to assessing for consensus sequences, SNP analysis, and basically a quality of mapping. Okay, so what did we see? So we obviously we did sequence a control sample. When you normalize the reads, these answers don't. These results actually don't change. We'll get to that in a minute. Why? So we sequence a control sample. Our actual fraction of reads that map to a reference genome is exceptionally low, and we sequenced a nickel strain, and we actually see less than 0.5% of our reads. And so we basically do not recover most of the genome. And when we actually, the direct comparison, so if we do the host depletion using NEB kit, which should technically pull out a lot of host DNA that's GC methylated, we actually don't really see a meaningful difference. We do see a bit more coverage, but it's still underwhelming. It doesn't seem to do a meaningful enrichment. So if we just take this control sample and do bait capture and specific whole genome amplification on it, we actually saw some really promising results. So for example, with the bait capture, what we see is we see that 80 to 90% of our reads are actually enriched. So this is really high enrichment. So we don't even need this step, because we end up getting with a relatively about 1 million reads, you can basically get the full genome. Why it's not 100%, we'll get to in a minute. This is a very promising result. The thing with bait capture, though, is it's relatively expensive compared to whole genome, the SWGA. SWGA, since this is about $15 a reaction that you add on to your full sequencing cost, whereas SWGA is just a couple dollars because you're just buying a polymerase and it runs at an isothermal. This is also a lot quicker. The bait capture approach will take you a day or two, whereas SWGA is just is within a day. But what we saw with the specific whole genome application is that we didn't recover full genomes. And so what's driving that is that when we look at bait capture, the really great thing about it is that we get relatively even genome coverage. So on the y-axis here, we have depth of coverage. So at this position here, for example, or across most of the genome, we have at least 100x depth, and a lot of positions were between the 1 and 200 base pair, 100, 200x depth. And this is for all the samples, and other samples we seek by bait capture, we see this very even genome coverage, which is very promising. When we look at these other methods, so this when we don't do these SWG or bait capture and this NEB GC pull down, you actually do not get very good genome coverage at all. What we actually see is that that most of the genome does not have very good coverage. Whereas and the same goes to control sample, it's not really different. You have a little bit more evenness coverage, but that was a stochastic effect. Now, when we look at SWGA, what we see is actually very uneven coverage. And we've nanopore sequenced this as well, and the results look identical, identical. So we actually have a problem with our SWGA primers, and we tried multiple sets. We still have the same issue. And this is the problem is that the primers are not capturing some areas particularly well. So we're gonna have to go back and design new primers. Because what we see is we actually end up having very uneven coverage. So we have these valleys and peaks, and these valleys here, we don't have sufficient depth to get the full genome. Whereas when we go back with bait capture, we do recover most of the genome, which is great. However, the one flaw that we have with bait capture right now, and this was done with Illumina, and we're using Agilent probes. And so Agilent is designed for Illumina sequencing. And one of the issues that we have with the Agilent probes is that they've designed it for very short read sequencing, so 1 to 200 base pairs. And most of our sequencing that we're using for this project is on the MiSeq, so 300 base pair reads or longer. And what we see is that because we're using short read sequencing, and then we're trying to optimize the protocol to kind of increase the length, is that we're getting gaps. And so if you remembered, I was talking about we have these Treponema, these TPR genes, these Treponema pallidum repeat proteins that are of interest for downstream analysis for rapid point of care tests. And what we do not recover that region well. So one of the areas that bait capture collapses is one of our regions of interest. Whereas when we go back to specific whole genome of interest, selective whole genome application, sorry, what we actually is we recover those regions fairly well, especially on nanopore. But when we do the short read sequencing with bait capture, we don't recover. So our assembly collapses in two places. It collapses on the 16S ribosomal RNA, which is repeated, although that's not of particular interest and relatively solvable with longer read approaches. Our TPR genes collapse. And what it ends up creating, it creates, when we do SNP calling, all those reads pile up on one set of those paralogous genes. And we get excessive SNP calls or these false positives, which are problematic if we want to use genomics for using this downstream. So what we really need to do is we need to kind of try to move our bait capture approach to long read sequencing so we can capture these areas. Yeah, so kind of to sum this up is we really actually do need if we want to, so there's the two parts of the program. If we want to do surveillance, this Agilent-based approach, so this getting nearly complete genomes is perfectly suited for that. If we want to start using the entire genome and so the genes of interest, like these TPR genes, we actually have to kind of separate our workflows into two streams. We have to create a workflow that we can reliably recover or solve our gap regions here to actually then get that information to mine for developing point-of-care tests that are based on reliable or high-quality genomic data. And then we'd have to create another higher throughput pipeline that you can multiplex because you have nice even genome coverage here, meaning you can actually pile a lot of samples together because 800 to 1 million reads will give you a high-quality, well-covered genome, which you get enough information to do kind of outbreak detection and analysis. And we have to create another pipeline to kind of look at using that information for developing like high quality genomes for other applications. Ideally, what we would want to do is if we can get bait capture working with the Agilent probes on nanopore, is that we can just recover these whole regions with long reads. So then we can just resolve this issue using nanopore sequencing. And so that's in progress. We're right now optimizing both the luminal protocol in terms of shearing, and then we're working on developing the nanopore protocols as well. As for the SWGA, we're redesigning the primers to see if we can reduce and get a more even genome coverage that would circumvent the costs created by bait capture. So I would just like to also acknowledge my references for this. So these images you saw earlier of syphilis, Center for Disease Control has a public health image library I'm sure many of you are familiar with, which is great. It has a lot of great images. I really recommend reading this paper by Joswal et al. It has talking about the pangenome. It does a very good summary of Treponema genomes. And then these two papers from Beal et al. One was looking at the global spread, and then the case example from England, and then Aho et al. For syphilis in Canada. And I just modified a whole genome application from NEB for my figure on that. All the other figures made from what I know. And then I would like to just acknowledge Public Health Ontario, which has supported my work on this, the National Microbiology Laboratory for supporting a genomics research development initiative on syphilis genomics, Samir, who's been very supportive. Other peoples of the micro and scientist team, Alex, Julianne, Sean, Angela, have been very supportive and helpful. And then as have Reza, Carla, and Sarah, and everyone else on the team at PHO that has supported my research to date. Thank you everyone for coming. If you have any questions and want an update, I presented a lot of our control. We're still working on finalizing some privacy so I can analyze some of the clinical data that we have waiting and that's in progress. But if you have any questions, please reach out and ask me after the talk if you have anything. Thank you, everyone.
Thank you so much, Tom, for that wonderful presentation. We're now going to move on to the Q&A segment of this event to address some of the questions from our audience. I encourage the audience to continue to enter your questions into the Q&A pod if you have not already done so as well. So one of the first questions that we have, and I think it's the one that kind of came up to a lot of everyone's mind when you were first showing the epidemiology of syphilis in Canada and abroad, is, what are the reasons and why are we seeing the rising rates more so in women in Canada compared to other populations?
Yeah, that's a good question. So no one. So the why is not very clear. There's been a lot of hypotheses put out there with things declining in sex education, less condom use, things such as PrEP, so people are less scared of getting HIV, for example, that this led to changes in sexual behavior that led to increased rates of syphilis. And then there's also things like perceptions of syphilis being restricted to some communities that once that escapes, that people aren't aware. There's the argument that once cases are low, or hypothesis that once cases were low, people weren't diagnosing it as effectively. So there's probably a confluence of factors, and I think public awareness might go a long way.
Thanks for that answer. Another question that we have in the Q&A pods, and I think you may have mentioned this, but perhaps some greater clarification, more explanation on, what the plans are in the future for this project is there a need for more samples, possibly clinical samples and whatnot? And also, what are the methods for how these samples are being collected and used towards this project?
Okay, so there is a need for more samples, but we have agreements in place to get samples from BC, Ontario, Manitoba. Right now, our big bottleneck is just we're finalizing genomic methods, so I'm waiting on results. So right now it's like I get results, analyze them, send them back to the NML, and then we keep refining the genomic methods. So our two bottlenecks is I need to redesign the SWGA primers because we ran through several rounds of primers and I tried combining the data and from multiple primer sets. So it looks like that gaps is just the methodological limitation of our primers. So there's optimization that is going to have to happen there. And then the other part of it is that so there's once that's done, we're hoping in the next month or two, we'll have a finalized bait capture method that will work well with Illumina. So right now we're just optimizing shearing, for example, on our control samples, because we don't want to burn through the clinical samples. We want to use that for the surveillance part with the agreements we have. And so going forward, once we have the genomes, we're just going to move forward with kind of understanding the pipelines to actually figure out what strains are circulating, and then start combining that with epidemiology. So that's the plan in the next few months. And then within the next year, we hope to have several hundred genomes that we can actually get a nice good snapshot of what we see circulating in Canada. And some of that we hope to have very high quality that can be used for the point of care test development. I can see so I hope that answers that question. If I missed anything, please follow up on that.
Thanks, Tom. So the work that you guys are working on towards those SWGA primers, remind me in the audience, those are those short length primers that were like 6 to 8 base pairs long, right?
Yes. Yeah. So I can. Why are they so short? So the whole genome amplification method, it's 529 DNA polymerase. It's just meant to create a bias because it's very quick. So like you can actually make them longer, 10 to 12 base pairs, but the longer you make them, and so what you're doing is just trying to bias because it's relatively quick and runs at a lower. Yeah, so I think longer primers tend to be less efficient. So you're kind of playing that length towards efficiency playoff there. And then someone also talked about the nanotrap microbiome B magnetic beads. We have not tried that yet. We can try that. That is something that we would like to do. Right now, what we did see is we saw such a really good optimization with bait capture, which you can dilute. So we did dilute our baits a lot, which is actually kind of nice with Agilent if you have lower your parasite load, the more you dilute, technically, the more you save on baits, which is kind of counterintuitive. But we saw with SWGEA and bait capture is that the host depletion, if there's like a cost-effective way to do it, we would pair it, but those two have been very effective on their own.
Thanks for that great explanation. I think one question that I can tie together with some of our Q&A pod submissions is the goal of this work towards possibly identifying vaccine targets or more so for looking at, surveillance and outbreak transmission cluster detection and possibly identifying potential missed cases utilizing this genomics approach and metagenomics work that you guys are doing?
So it's both. So my interest in the project is more on the surveillance side of thing. Venkata is leading the vaccine point of care test, sort of like, is it possible to use the genomic data for that? I am personally interested in kind of like what is behind the increase, what strains are circulating. Can we use this to do some public health interventions? How effective will education be for that? So there'll be and part of it would be then to create these pipelines and protocols for public health units to use right away for local public health use. And out of that, you get research as well, naturally. But yeah, a big goal of it is to create that kind of that out of the box, ready to use approach for public health.
Another question that we have in our Q&A pod that's similarly aligned to this, and I'd like to kind of expand it more of my own personal question is, are you aware of any other jurisdictions, whether that's within Canada or, abroad globally, that are doing syphilis WGS or utilizing genomics for public health purposes yet? Or are they more sort of in a research and development stage still?
So I've only seen research on it. I'm not quite sure what other jurisdictions are routinely implementing it. I think there is interest in the UK in doing that, but Australia and Japan have recently, in the last few years, released surveillance papers with a high number of genomes. So I think that's something that's probably in the works, but I don't know of anyone explicitly routinely using it as a surveillance program.
Thanks for that answer. I think one question that we have in the Q&A pod that's probably something that's, folks might be wondering is, why did we choose to start with syphilis rather than a different pathogen? Is part of it because it's so hard to grow actually in a lab, like you mentioned with the rabbit testes, or is there other purposes, associated with the genome of it and whatnot that makes a really good candidate for looking at it from a metagenomics approach?
Yeah, so that's a good question. So what's driving this project is the increase of cases we're seeing in syphilis that we kind of want to, and the other challenge with syphilis in general is that there is actually, the genome itself doesn't have a lot of diversity. So if you think of like TB, it doesn't have a lot of genomic diversity, but we can culture it. The challenge with syphilis, it doesn't have a lot of diversity. So choosing small, short molecular markers, it's kind of what the problem we have with TB is that you underestimate diversity, so you kind of over cluster things. So we really want to understand what's going on with something that's having this like 758% increase in females, for example. We kind of need higher resolution methods, and genomics offer that. The challenge with syphilis, and this is actually kind of a nice thing to develop, is that there's public health interest in this and actually kind of going through and testing different metagenomic approaches to capturing pathogen genomes can be a good case example. So if we run into something else where we can use these methods, we're more familiar with them. the driving factor behind it is trying to understand what's going on with syphilis. It just also happens to have a interesting lab challenge to see, can we use these metagenomic approaches that can be very inefficient in the future more efficiently. So when we think of like when Mpox first had the outbreak, we're just sequencing those whole extracts. And so you're trying to do this, get these massive amounts of data to just for like less than 1% of what you have. Is there a way we can do that in the future, but just in a much quicker, more effective manner? Would be like an added benefit of working with this. And since we can't really effectively culture it, it's good, but we can get good controls for it. Because like you said, you can get nickel strain cultured in rabbit testes. So you can have a relatively high quality control sample that you can play with and work with to optimize, and then you can apply to clinical. So it's a good example on the lab side for that as well.
One question that I wanted to ask you, Tom, is you had mentioned that, penicillin does remain sort of the main treatment antibiotic for syphilis, that it's highly effective. But in cases where patients may have allergies or don't tolerate penicillin and they would use macrolides or doxycycline, which seems to be the case that we are seeing kind of more resistance to syphilis now with these agents. And you had mentioned that, genomics can be utilized towards AMR prediction for syphilis in the future and work towards that as one of the future goals of the project. How does, syphilis develop, or the Treponema pallidum develop resistance to these agents? Is it more so within the genome and mutation there, or is it actually acquisition of exogenous, mobile genetic elements and whatnot? And sort of what's the approach from a genomic standpoint of how you guys would be identifying, and predicting resistance to the drugs then?
That's a very, very good question. So let's so I guess it's easier to talk about the genome. So it being not resistant to penicillin, I think it's just the treatment method's very strong and effective right away. So it's whereas something like macrolide resistance was, I think it's oral. So but when we look at the syphilis genome, so when you talk about pangenomics, for example, there's talking about whether a genome's open or closed. So if a genome's closed, the more genomes you sequence, you're not going to really see new information. What we see with syphilis is that it's still technically open, but there isn't, the more genomes you sequence, you don't really add a lot of new genetic information. So there doesn't seem to be a lot of exogenous DNA in there. But we did note, but there is, within the pangenome, some of those pathogenicity islands and genomic islands were introduced from transposons or viral elements. So that is possible, we just don't really see it. So there isn't a lot of like diversity in terms of gene content within syphilis, which is why we don't really see a lot of resistance. So we don't see any resistance to doxycycline that we know of, but we don't really, because a lot of the few doxycycline resistant genes that we know exist out there don't exist in Treponema. So that will get us to the predictive problem. But for something like macrolide, a lot of these mutations are well established. So that's basically just matching the mutations to a known database. So we can track that well, and what we do know from the global data is that macrolide resistance is prevalent. It's not fixed. There's still sensitivity within syphilis strains. And the prediction part's a hard part. So that is something we're still trying to work out. So that's something Venkata and I will collaborate on. So Venkata does a lot of machine learning to see if there's a way we can predict novel resistance. And that is actually a very hard problem that I don't think we have really figured out a way to find, right? Because you also can't readily culture it to test it. So there's a lot of issues around like developing that method, particularly with syphilis. You might have to use another model to then apply to syphilis, but that's a very good question. But as for the genome, there's not a lot of diversity. So we don't see a lot of we don't see any resistance to penicillin, and that could just be how the treatment takes place versus anything special than anything magical or anything special about or different about Treponema relative to other pathogens.
Thanks for that really thorough answer, Tom. One question from our Q&A pod in relation to, its potential uses of syphilis genomics for surveillance and public health, possibly contact tracing is, what methods do you envision will allow you the WGS data to be correlated or calibrated with, case contacts or specimens and, differences in time in between when transmission occurs between individuals, and how is genetic relatedness for syphilis, how does that differ for other organisms in terms of does it mutate quickly, or is it relatively slow that genetic relatedness can be used to support contact tracing from an epi and public health standpoint?
That's a very good question. So I'll start with the genome part again. So we won't have a lot of metadata, so we'll associate what metadata we have. So where they live, age. We'll start with a very simple metadata and see what other additional information we can get to associate with the genomic data. Because genomic data is only really useful, as we saw at the ending example, when it's combined with epidemiological data. So the genomic data can really give us well-supported clusters, and that once that's associated with epidemiological data, that can actually give us insights. But when it comes to the genomic data, syphilis is very slow evolving. So actually relatively few SNPs can give you a very confident cluster, sort of. Because this is one of the challenges with something that's very slow evolving. You have to be careful about how to interpret the data. It's different than things that evolve very quickly, is when you can be less confident. There's the same problem with a few amount of SNPs, but once you have the whole genomic data, you can get relatively robust cluster information that you can then combine. So you're not worried so much about like convergent evolution, because I think in several cases, if you look over a few-year period, you can see one strain not show any variation within a 2-year period. So that's both a blessing and a curse with syphilis. But yeah, to make sense of the data would have to be started to be combined with things like epidemiological data to kind of understand. So do we see different strains circulating among different communities, right? So why do we see a 758? Is that being driven by a particular strain, or is there a bunch of different importations and local spreads? That's something that we need epidemiological data to understand.
Thanks for your answer, Tom. One question that's come up is, and whether or not you guys have had a chance to look at this, is what's the differences in recovery of nucleic acids among the different specimen types, and whether or not you can anticipate some challenges that might come with, with actual collection of clinical specimens and, those differences in collection methods and stages of infection and disease that you guys might have predict will be some difficulties that will come up with the genomics, and whether or not you will be able to get enough of your pathogen DNA or not.
Yeah, so that's a great question. So right now we're just doing primary ulcer swabs. And so that tends to have the highest parasite load. Downstream, we'll start looking at other sample types. As a general rule, so for all the samples we looked at, and even our control samples, is we do qPCR to do copy numbers, and you actually get, even from the primary ulcers, you get a huge range in parasite load by a couple orders of magnitude. What we did notice is with the bait capture and SWGA, even with these relatively low copy numbers, they work very well at enriching either way. So that's actually a really good plus for the adjuvant bait capture probes, is because the less parasite you have to actually end up diluting the probes to get an optimal result, and it seems to work surprisingly well. And same thing with the SWGA. So we had a couple samples. We have the qPCR results that I'm waiting to get the results to analyze, but we do the qPCR before we do the enrichment, and then after we do the enrichment, and you'll see more several orders of magnitudes increase in copy number per microliter. So you'll go from something like 2,000 copies per microliter of Treponema to about 20 to 60,000 copies per microliter. And that enrichment seems to sequence out quite well. And once you have the post enrichment to the human, the RPP30 copy number drops substantially by several orders of magnitude. So you kind of have those two levers with the target enrichment that works really well. What I'd really like to see is how the differential lysis works, but that's something that's on pause for now. And try other capture methods like those other beads that were mentioned. So
Thank you so much for your answers, Tom. We're just about at time. So as we wrap up today's PHO Microbiology Rounds, I'd again like to thank Tom Brockman for presenting a wonderful presentation. And I also like to thank everyone who joined us for today's webinar. You can expect a brief and anonymous PHO Microbiology Rounds survey for today's session. And please try to complete this, as this does help us improve our programming. Lastly, to access past PHO presentations and to view confirmed and upcoming sessions, please visit the PHO website, head to the education and events, and then click on presentations. Thank you, everyone, and have a wonderful day.